Hi everyone, welcome to Bioinformatics with Vaishnavi. In this video, we will understand next generation sequencing, also called NGS, and we will compare it with the traditional Sanger sequencing method. This video will help biotechnology and life science students understand why NGS replace traditional sequencing techniques. Before diving into the topic, let me introduce myself. I am Vaishnavi, a biotechnology graduate working as bioinformatician in India, and on this channel, I simplify bioinformatics for you. So let's start. NGS stands for Next Generation Sequencing. It is a high throughput sequencing technique, which means it can sequence millions of DNA and RNA fragments simultaneously. Unlike older method, NGS allow us to generate huge amount of sequence data in a single experiment. Now we will see why is NGS important. See, NGS is important because it has completely changed biological research. It is widely used in whole genome analysis, transcriptomics like RNA sequencing, epigenetic studies, precision and personalized medicines. Without NGS, many modern research and clinical studies would not be possible. आपके दिमाग में भी ये क्वेश्चन आया क्या कि पहले कैसे सीक्वेंसिंग होती थी अगर एनजीएस अभी नई टेक्निक है तो चलो देखते हैं Before NGS, DNA sequencing was mainly done using Sanger sequencing. Let's briefly understand how Sanger sequencing works. See, Sanger sequencing is based on the chain termination method. It uses special nucleotides called DDNTPs, dideoxynucleotides. During DNA synthesis, when DDNTPs incorporate, the chain stops growing. This creates DNA fragments of different lengths, which are then separated on electrophoresis to determine the DNA sequences. Okay, now we will see why NGS better than Sanger. Compared to Sanger sequencing, NGS offers many advantages. NGS can sequence millions of fragments at the same time, which makes it high throughput, right? It is scalable, more sensitive, has higher discovery power, and becomes cost effective when large datasets are analyzed. That is why NGS is preferred for large scale studies. Now we will see why we still use Sanger sequencing. हम NGS यूज करते हैं लेकिन आज भी for validation we use Sanger sequencing. Why? Even though NGS is widely used, right? Sanger sequencing is still very important. Sanger sequencing is known as for its high accuracy, which is why it is considered as golden standard for validation. It is also suitable when we need to sequence small DNA regions or a few samples. So common NGS platforms, there are several ones. So most common are Illumina, which uses sequencing by synthesis, Iron Torrent, PacBio, Oxford Nanopore. Each platform has its own advantages and applications, which we will discuss in future videos soon. This was a simple explanation of next generation sequencing and how it compares with Sanger sequencing. If this video helped you, please like, share, and subscribe to Bioinformatics with Vaishnavi. Thank you for watching, and I will see you in the next video. Bye bye.